Now let's discuss intestinal protozoan infections. So the protozoan parasites causing intestinal infections are intestinal amoebae, so Entamoeba histolytica, flagellates, so Giardia lamblia, and opportunistic intestinal coccidian parasites, so Cryptosporidium, Cyclospora, and Cystoisporidia. Next, intestinal amoebiasis. So amoebae means change. So they are single-celled protozoan parasites that constantly change their shape because of an organ of locomotion called pseudopodium. Based on the habitat, they are of two types: intestinal amoebae and free-living amoebae. So Entamoeba histolytica infections. It causes amoebic dysentery and a wide range of other amoebic invasive diseases such as amoebic liver abscess. There are three other species of Entamoeba such as Dispar, Moshkovsky, and Bangladeshi, which are similar to E. histolytica. So it is worldwide in distribution but more common in tropical and subtropical countries. Largest burden of the disease is seen in tropics of China, Central and South America, and Indian subcontinents, and it is the third most common parasitic cause of death in the world. Moving on to the morphology, first you have the trophozoite. So obviously you have the pseudopodium. Because of it, it is free moving amoeba. And then you have within the cell RBCs, WBC, food vacuoles, and also a nucleus. So central karyosome surrounded by peripheral chromatin. Next, you have the cysts. So you have the precyst with the nucleus only. Then you have the uni and binucleated and then quadrinucleated cysts. So in the uni and binucleated cysts, you have the nucleus, okay, according to the number in the name. And then you also have the glycogen mass for food and chromatoid bodies. You don't have chromatoid bodies, okay. Now moving on to the life cycle, first humans take in the cyst, the quadrinucleated cysts in food and water contaminated with feces. So then there is excision in the small intestine. Trophozoites are released and they are carried to the large intestine and they colonize the large intestinal mucosa. They again form cysts which are released in the feces, and mature cysts are again acquired via food. So trophozoites also penetrate the intestinal wall and are carried to extra intestinal sites. So virulence factors, what are they? You have the amoebic lectin antigen. Trophozoite form is the invasive form, okay. So you take in the cyst, send out the cyst, but infection is by trophozoites. So amoebic lectin antigen, amoeba pore, cysteine proteases, and hydrolytic enzymes, and collagenases. And so pathogenesis is basically, so it colonizes the intestinal mucosa, the trophozoites. It adheres to the mucus layer and large intestinal mucosa by Galnac lectin molecules. Then it forms flask-shaped ulcers and then invades the large intestinal wall to migrate to extra-intestinal sites. So here you can see the flask-shaped ulcers, okay. It adheres to the mucus, then to the mucosa, and then causes flask-shaped ulcers. How does it do that? Galnac molecules. So clinical manifestations, males and females are affected equally. Majority of infections result in asymptomatic cyst passers. Remaining develop intestinal amoebiasis. So clinical manifestations are dysentery, appendicitis, amoeboma, etc. So here differences between amoebic and bacillary dysentery. Remember A. So amoebic, there is A. So A for acidic, A for offensive, okay. Next, A for dark red, okay, and then A for large amount of urine. So if you are excreting out large amounts of feces, then you need to go for less number of motions, so 6 to 10 per day. But in bacillary, if you are excreting small quantity of feces, you go for more frequency, right? So large number of motions. Next, it is also not adherent to the container. Next, moving on to the type of ulcer. Ulcer is deep and the margins are ragged and undermined. As you can see, they're ragged and undermined, whereas it is uniform and shallow in bacillary dysentery. It is just like this. So intervening mucosa is normal, but in bacillary dysentery, they do a lot of action, right? So a lot of mucosal cells are inflamed, so that is why polymorphonuclear cellular response is seen, whereas in amoebic dysentery, it is mononuclear. So you can also see RBCs and clumps in amoebic dysentery. Amoebiasis, so eosinophils and charcot-laden crystals and pycnotic bodies are present, okay. Moving on to the lab diagnosis, you can see you can do stool microscopy by wet mount, etc., to detect cysts and trophozoites. Histology, you can take intestinal biopsies to see the trophozoites on the intestinal mucosa, and stool culture as well. So you can do stool antigen detection by ELISA, so lectin antigen, and you can do serology, ELISA amoebic antigen and amoebic antibody, and isoenzyme analysis. So molecular diagnosis, you can do real-time PCR. All these are the pictures. Okay, this is the triage parasite panel positive for Entamoeba histolytica. So asymptomatic carriage treatment helps in reducing the passage of cysts in stool and thereby prevents the disease transmission. So luminal agents are used. So iodoquinol for 20 days or paromomycin. Remember parvaledu or purgative diarrhea, right? So paromomycin for 10 days. And again in intestinal amoebic dysentery, you use luminal agents like paromomycin and again use metronidazole. Other measures include fluid and electrolyte replacement and symptomatic treatment. Preventive measures include avoiding ingestion of food and water contaminated with feces and treatment of asymptomatic carriers. Amoeba morphologically resembling Entamoeba histolytica. So we have Entamoeba dispar, Moshkovsky and Bangladeshi, which are similar. So dispar is non-pathogenic commensal which is found in the human intestine. Moshkovsky is sounding Russian, right? So it is a free-living amoeba found in polluted water. Okay, it may occasionally cause non-invasive diarrhea in infants, and E. Bangladeshi has been recovered from asymptomatic individuals as well as those with diarrhea. So how do you diagnose and differentiate it from Entamoeba histolytica? Only Entamoeba histolytica has RBC inside the trophocytes. It's a marker of invasiveness. And also you can do PCR, which is the most reliable method. You can amplify the small subunit rRNA gene and also lectin antigen detection in stool. Non-pathogenic amoeba, the prototypic example is Entamoeba coli. You also have Entamoeba gingivalis. So what are the differences between histolytica and coli? So in histolytica you have RBCs and WBCs, and also the trophocytes, the cytoplasm is clearly differentiated into ectoplasm and endoplasm. And also the nucleus, you have a central small point-like karyosome surrounded by nuclear membrane which has fine chromatin granules. But here you have a large and eccentric karyosome and coarse chromatin granules are present. And Entamoeba histolytica, it is obviously having pseudopodia, so it is very active in motility. And also basically the nucleus is the same as trophocytes in both. The nucleoli are more in coli. Okay, 1 to 8. Here it is 1 to 4. That's it. Moving on to intestinal flagellate infection, so we'll be talking about giardiasis. So Giardia lamblia causes that. It is also known as G. intestinalis or G. duodenalis. It was first observed by Anton von Leeuwenhoek while observing his own stool sample. It is worldwide in distribution. It is considered as one of the most common parasites, both endemic and epidemic intestinal disease and diarrhea. So it is common in warm climate of tropics and subtropics, and genotypes are A and B. Then the morphology. So first you can see it is a teardrop shaped, and you can see two sucking discs are present. Within each sucking disc there is one nucleus and one central karyosome, and there is an axoneme and several flagella are present. Okay, here you can see that the cyst has a cyst wall, the axoneme again, and 4 nuclei, not 2. Okay, so the life cycle, Giardia completes its life cycle in one single host. The infective form is cyst, not trophocyte as in the case of Entamoeba. So basically you take in the cyst and pass out the cyst again. Man acquires the cyst by ingesting it, and then excistation is there in the duodenum. Trophocytes are released. They multiply by longitudinal binary fission. They adhere to the mucosa, but they do not infect the mucosa. They just feed on mucous secretions. They do not invade it. The trophocytes are released into the lumen. They go into the jejunum, encystate, and then cysts are passed out in the feces, and the cycle repeats. So infective doses, very few cysts are needed. 10 to 25 can initiate the infection, and children are more commonly affected. So pathogenicity, the pathogenic form is trophocytes, infective form is cyst. Okay, so trophocytes adhere to the duodenal mucosa, cause disruption of the intestinal epithelium that leads to increased permeability and malabsorption. Trophocytes do not invade the mucosa, they just adhere to it, cause its disruption, and feed on the mucous secretions. So malabsorption is caused because they just adhere to the mucosa and feed on its secretions, right? So malabsorption of fats, steatoria, foul-smelling, profuse frothy diarrhea. So malabsorption of sugars, so disaccharide deficiencies, and malabsorption of vitamin A, B12 and iron, and also protein-losing enteropathy. There's also antigenic variations. So Giardia undergoes frequent antigenic variations due to a cysteine-rich protein on its surface called variant surface protein. Now clinical features, so there are asymptomatic carriers, most of them are asymptomatic, and then acute giardiasis, incubation period varies, and chronic giardiasis, it may present with or without a previous acute episode. So lab diagnosis, stool examination is the gold standard test. So it detects cysts and trophozoites in the stool, but if you see pus cells and RBCs, it suggests an alternative diagnosis because pus cells and RBCs are never seen in Giardia samples. So you can also use the gelatin capsule enterotest or string test. So basically there's a string with a gelatin capsule and a weight on one end, and then you make the person swallow it, and there's an end of the thread which is attached to the person's cheek. Okay, so that is used. And then antigen detection in stool, you can do copper antigen detection, antibody detection by ELISA, and culture. Also, Diamond's media is used for Entamoeba histolytica. You can do PCR also. And treatment is by using metronidazole. But again, some AIDS patients, giardiasis is often refractory to treatment. There is prolonged therapy with metronidazole in those cases, but metronidazole resistance has been reported in giardia. Next, prevention is by improving food and personal hygiene and boiling or filtering of potentially contaminated water and treating asymptomatic carriers.